This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 86 for June 11th, 2010. Hi, everybody. It's Vincent Racaniello, and this is TWiV, your weekly dose of viruses. And today we're coming from San Francisco. I'm with my co-host, Rich Condit.
Hello, Vincent.
Looking at you again.
Yeah. Were we actually sitting side by side last time we did this? Yes.
We were. And
right.
You said this is pretty strange.
On the other side of the country.
Here we are. So Rich and I are working out here in San Francisco, and we're visiting a colleague of ours, Eric Delwart. Welcome to TWiV, Eric.
Hello, and welcome.
Eric's at the Blood Systems Research Institute, which is right in San Francisco.
Correct.
That's where we are. We decided to come visit Eric because we've been talking about his work for a long time, as long as TWiV has been on. So since Rich and I were out here, we thought it'd be a great opportunity. And I got to tell everyone, we had a great, Alan, going through all the hosts, Eric gave us a great tour of various places in San Francisco. Land's End, where you can see the coast and the Golden Gate Bridge.
Golden Gate Bridge, beautiful view of the bridge.
And Rich is crying because he was brought up here, right?
Right. It really tugs at my heart, this place. It's wonderful. What is it? I left my heart in San Francisco.
That's right.
I won't sing for you.
You probably did. And then we had lunch on the coast on Route 1.
Yes, by the ocean front, the windy part of San Francisco.
Great. And of course it's Friday, so everyone's getting in a Friday mood here in San Francisco.
And it's a beautiful day. Nice sunny day.
It's gorgeous out.
Gorgeous.
But now we're down to work here at the Blood Systems Research Institute, which is where Eric works, and we're going to find out all about what he does and how he does it. Starting with, what is a Blood Systems Research Institute?
BSRI is the research arm of Blood Systems, which is a non-profit blood bank, which is actually the second biggest blood bank collection and testing agency after the American Red Cross. They have about, I think, 20-25% of the blood business, and they're quite successful, and they have decided to fund a research institute to ensure the safety of blood, be it through the threats of viruses or immunological reactions. So our overarching mission here is to keep the blood safe.
So when we walked in, we passed areas where blood is being collected and stored.
So half of the building is a regular blood bank. People sit down, give blood. Blood is processed and shipped out for further testing. A lot of blood now is extremely safe because they're testing for nucleic acids of all the usual blood-borne viruses like hepatitis B, HIV, and Hep C. So it's this is done. This is centralized, and the blood actually flies out every day, a little aliquot of it, while the main blood product remains local. And when the data comes back a few days later, it's deemed safe or not, and then distributed to hospitals.
So do you have a role in that process?
I have nothing to do with the regular running of the blood bank, but I'm involved in the research section. Okay.
So I guess I'd like to talk about that, so exactly what you do, but I want to find out how you got here. What's your path, your career path of coming here? I know it's a wonderful circuitous pathway, but I want you to tell our listeners where you started. You were born in the U.S. but grew up in Switzerland, right?
Correct. I was raised pretty much in Europe and in Belgium. I got my undergrad in England, University of Newcastle. Then I started a PhD program in Geneva, which I abandoned to come to California. It was hard to resist.
So you left before getting a PhD?
Yes, I ended up with a master's, and I went to work for Genentech, which was at the time a already well-known but still small company. And there I really felt the excitement of discovery. I was, very peripheral, but I saw the discovery of the cloning of factor VIII, TPA. But then I realized as a technician, your options are limited. I'm sure I would have done very well if I'd stayed at Genentech, but if you wanted to sort of, control your destiny, you really did need a PhD. So
How long were you at Genentech?
Just a year and a half.
Okay.
And I then went to the University of Wisconsin in Madison, great place, and I got my PhD working on retroviruses, an obscure avian retrovirus in the lab of Antonito Panganiban, who was himself a newly minted faculty, sort of on the, he had himself been trained by Howard Temin. And so I worked there on receptor interactions of retroviruses. And my postdoc, I continued, then HIV came up onto the scene just about the time I was finishing my PhD. I dabbled in HIV, and I then continued my postdoc at Stanford with Dr. Mullins, Jim Mullins, where I sort of stumbled onto a very simple assay to measure genetic relationship between DNA strands. By simply melting and re-annealing them and running them on an acrylamide gel, you can eyeball how different the strands are. The more distinct they are, if they can still re-anneal, the slower they migrate on a gel. And it was quite a nice assay because at the time sequencing was still expensive. And the WHO thought it would be useful to train people in developing countries where they had the AIDS epidemic, HIV epidemic, to analyze their strains in different countries. We wanted to know the subtypes of HIV in Thailand, in different countries in Africa. So they had a workshop where I was part of that, and I would fly out and train people on how to do PCR and run acrylamide gels, basically. And that was fun. I got to see Thailand, Bangkok, and then Entebbe in Uganda. And we also had a workshop in Belgium.
So when was this? What year are we talking about now?
82, 83.
Okay.
Beginning of the AIDS problem.
Sorry, no, this was later than that. I mean, HIV was isolated in 83.
Yeah.
So this is more like 86, 87.
Okay.
So 86, I think, 85, 86 was when PCR came on the scene. Is that right?
Sorry, I got my dates completely wrong here.
It's okay.
No, because I was doing my PhD then. This is more like the early 90s.
Okay.
And then after my PhD in Madison, I was at Stanford doing the sort of genetic analysis of HIV. Then I moved to the Aaron Diamond AIDS Research Center, which was a booming little research institute in New York, which was on the cutting edge of AIDS and HIV research. The protease inhibitor had just kicked in. Therapy was looking quite good. There was talk of virus HIV eradication, which since has sort of disappeared. And I continued to work on HIV for a few years.
So there you were independent?
Yes. So I had it's called tenure track.
Yeah.
And after a few years, the call of California sort of came back. And
I can relate to that.
And I had worked, while a postdoc, I had worked with Mike Bush, who was the director of our research institute here. And so I contacted him and says, come on over. You know, you've got some grant money. You work on viruses. We're always concerned about the safety of blood. So I came here back, and I've been here for about 10 years now. With an appointment, one of those honorary appointment adjuncts at UCSF. But my lab is in the Blood Systems Research Institute. And I continue to work on HIV at the beginning, but then, you know, I think it's healthy every 10 years or so to sort of start to dabble in other fields. And so I work quite a bit on hepatitis C, which has many of the properties of HIV in terms of its evolution, its genetic diversity, all happening very quickly. I mean, within weeks, literally, you can see the viruses change within people. But then there was this paper I saw in the PNAS, a rather sort of obscure sounding paper about a new way to identify new viruses, which when I got into the materials and methods, I realized how simple this was. And the idea was just take any biological sample and you can filter out the big particles like cells and bacteria so that the viruses would go through the filter, take that. And the key here in that paper was to remove the free-floating DNA and RNA that is always present in fluid. I mean, human plasma is full of human DNA. If you get rid of that, what you end up with is basically viral particles with their nucleic acid. At that point, simple thing to do was to extract and do what's called a random PCR, which is a bit of an oxymoron, but it's basically PCR that amplifies everything. And it's based on the degeneracy of the primers, which will sit everywhere across all nucleic acids. So I thought, I want to try that. We were lucky in San Francisco. There are a lot of people collecting plasma sample from high-risk groups. And Rick Hecht, a professor at UCSF, said, I have febrile blood from people that we thought had HIV acute infection, but did not. So maybe there's something else is infecting them. And literally within a few weeks, we had stumbled onto our first virus. It was a parvovirus, which is the same family as B19, which is another human parvovirus. And at the time, it was the second human parvoviruses. And I thought, this is just too good to be true. There's just so many undiscovered viruses out there. So I started redirecting a lot of my effort towards viral discovery. And the way we get interesting samples is basically I read people's papers, usually in clinical virology journals, where they test their samples for the usual culprit. But in a large fraction of cases, they can't find any viruses. So these are the samples that I asked for. So I'll email people, say, do you want to collaborate? We'll do this. It's called Metagenomic, which is basically let's sequence everything in sight and see what it looks like using deep sequencing. Deep sequencing is just high throughput sequencing. You just take all the nucleic acid and either do it the old-fashioned way through Sanger sequencing after you subclone, or now with these new technologies, you can literally generate millions of reads within a few days for a lot less money than you can with Sanger sequencing.
Why is it called deep?
Because you look deeper into the population of nucleic acid. If you do a superficial sequencing, you'll end up sequencing what's most common in that mixture. But if you go deep, you start picking up other stuff, lower frequency nucleic acid. For example, if you have a lot of human DNA or other virus in there, you're not going to pick up the rarer strains unless you go deep.
The technique does involve an amplification step, however.
Correct. Deep sequencing is also what people use when they're sequencing human genomes. I mean, everybody now, is trying to understand human evolution and some diseases through sequencing human genomes. There's so much DNA you don't need to amplify. You just take a couple drops of blood and you get enough human DNA to sequence. But when you're looking for new viruses, there's so little nucleic acid there that you need to amplify it. And also, most viruses are actually RNA viruses. So you have to reverse transcribe that nucleic acid and turn that into DNA before you can sequence it.
So when you do this, do you like split up populations and have a reverse transcribed population and one that's not?
Actually, we do. We'll take an aliquot, we'll put some aside in case something interesting comes up, and with the rest, we'll split into sort of RNA fraction, and we'll get rid of the DNA by just enzyme treatments. And then we'll do vice versa. We'll have a DNA only where we amplify without the RT step, the reverse transcriptase. So then we have, if there is anything in the sample, and most of the time there isn't, we'll know whether it's a DNA origin or RNA origin. But we can usually tell when you have a new virus, if it looks like a DNA virus, it's probably going to be a DNA virus. And same for RNA.
So if you're looking for in a sample, you don't know what to look for, an RNA or a DNA virus, right?
Correct.
So let's say the amplified portion where you take RNA, you copy it to DNA, and then you take random primers or degenerate primers and amplify. Then you subject that to massive sequencing where you go many. A read is simply a stretch of nucleotides that you get from the machine. So a
sequence read, depending on the technology you use now, with Sanger you can sequence about up to 1,000 base pair long. With the very highest throughput methodologies, it's about 75, maybe 100 base pairs. We use another sort of next generation sequencing called 454 pyro sequencing, where the reads are about 400 base pairs on a good days, and that's long enough. When you take all these millions of reads, 400 base pair long, when you put them through pretty heavy-duty computing analysis, you basically compare them to all the known viruses in the databases. And if you have a significant similarity in terms of sequence, you can usually tell whether it's a new virus or not. Sometimes there's gray zone sequences, could be, but statistically it's not very significant. Sometimes it's in your face, you got something new. And you can go from a sample to a new virus pretty quickly.
So how many reads would you do for a sample typically?
It all depends on the concentration of virus. For example, if we have tissue culture material where people will contact us and say, look, we have something that grows in culture and we know it's there because it's killing the cells. but we don't know what it is. There, the virus is so concentrated, you really don't even need next generation sequencing. You can just subclone into a plasmid and sequence 10 inserts. If you don't know, and most of the time we don't know if there is any virus or what the concentration is, then we go a bit deeper with next generation pyro sequencing.
A bit meaning?
So right now we can tag each of our samples so that we can recognize them, and we pool them. So it is still a bit expensive to do a next generation sequencing run, anything from $5,000 to $10,000. So we can't just look at every sample at that depth. It's too expensive. So we'll pool about 50 samples, each tagged with different primers at the extremities, so that we can recognize where the sequence came from. And then we keep aliquots of those 50 samples in case something comes up. Sometimes we get the full genome if the concentration is high enough, and that's very satisfying. That's almost a publishable unit at this point, if it's an interesting enough virus. Sometimes we just get little fragments of it that are over the most conserved regions. And then we will basically link up these fragments of viral sequences until we get the full genome. Sometimes if it's a circular DNA virus, it's quite easy because you get a little fragment. You do what's called inverse PCR. You can go around the circle to generate the rest of the sequence. If they're linear, it's a little more tricky, but there are ways to chromosome walk, in effect, to extend the known viral sequences. You get a foothold. You've already cloned and sequenced a bit of it. You kind of do extensions on both sides of that foothold to get the rest of the genome.
So if there's a virus in a sample that has nothing in the database, a totally novel sample, can you recognize that?
No. That's what we like to call the dark matter. And about 30% of sequence information from this sort of studies, whether it's virus hunting or environmental, deep water, sort of let's see what's there, bacterial sequences, there's no homology. The sequences you derive don't look like anything else that's known or that's annotated in GenBank.
How do you know they're viral then?
We don't. But we suspect there are viral families out there that have still not been identified that would not be recognized by these similarity searches. And we are struggling to find ways to identify those. I mean, the best way would be to be able to grow this virus in culture and then have high titer and show EM pictures, for example. But that's very hard. You don't know what the tropism is. You don't really know what the host is. So the way we sort of grappling with this is to see whether we can build large contigs, whether the nucleic acid is concentrated enough to yield bigger pieces than just these single reads. And that's a landmark of something that's at a high concentration, as you would expect a virus to be. It also reflects a small genome, as viruses have. So, but still, you end up with a larger contig, let's say a few thousand base pair, that still doesn't look like anything. And is that a virus or not? So one way to increase your confidence that this may be a new virus family is to search for this particular sequences in other individual. I mean, is this a common infection? And then our hope is as you grow these sequences, eventually you will recognize some kind of viral signature, maybe a very weak protease domain, reverse transcriptase, RNA-dependent RNA polymerase, some kind of a hallmark gene of viruses.
There could be viruses without such signatures,
yes,
though, right?
And we will, I'm sure we're sitting on many viruses right now that we don't recognize.
Give us a number. So how many, we know a few thousand viruses right now, total human, plant, insect. How many you think remain to be discovered?
Well, among humans, we probably know a fair bit of human viruses. If you take all the mammals, we probably know less than 1%. So many mammal species. I mean, we just finished a study of bats, bat poop, really? that we got from actually Point Reyes up in Marin County, from another collaborator from University of Boston who went and collected bat feces or guano. And there we found all kinds of new viruses that weren't known before. We also found a lot of ingested viruses from their food, a lot of insect viruses because these were bats that eat insects, a lot of plant viruses, and these are probably plant viruses that the insects had eaten and then ended up in the gut of the bat.
Sure, sure.
So we know very little about the virome, the animal virome. And when
the dark matter is going to be really interesting.
Most interesting, right?
Yeah, I mean, it's also nice to find viruses that you can recognize, especially if they're human viruses, especially if they're related to pathogens, like the virus related to the Picorna and part of the Picornaviridae family. I mean, Kapoor, who basically started this, found that it was a very common infection that had been overlooked. Is something like 20% of kids at any given time are excreting this virus.
Globally or just in
at least in developing countries. Okay. We actually know it's in the U.S. because if you look at sewage water.
Really?
Mia Bridbard up in Florida actually sent us some sewage water, extracted nucleic acid, and we tested it for COSA and other viruses we found in developing countries. If you look hard enough, these viruses are global. Mm-hmm. So it's nice to find a known virus related to a pathogen like COSA virus, in the sense it's distantly related to the polio and the enteroviruses. It's extremely diverse. And like many viruses, it's probably pathogenic in only a fraction of infections, maybe 1 in 1,000. So it's very hard to go from identifying a new virus to proving that it's a pathogen. It's sort of an issue now with this viral discovery. A lot of people are finding new viruses, but only a small fraction of these new viruses are actually now shown to be pathogens. There may be a lot more than maybe end up pathogens, but if they're pathogenic only rarely, you really have to do large studies to associate them with
couldn't
disease.
It be that most viruses of humans, just to make it narrow, are in fact not harmful to us?
It's a good argument that we have already found the real nasty ones because they cause acute symptoms.
They're obvious. Yeah.
And people pay a lot of attention. But there's also a lot of diseases out there that large fractions are unexplained. Diarrhea, respiratory illnesses, a lot of clearly there are viruses causing these that are not yet been identified, and some of which have been recently identified. A lot of autoimmune diseases may be induced by infections, and you end up with diabetes or rheumatoid arthritis 10, 20 years later. Some cancers, I mean, the Merkel cell polyoma virus is a nice example. There may be other cancer-inducing viruses. These will be tougher to find because when you're looking at not biological fluids like plasma or even stool, where you basically we look at the stool supernatants after we filter it. When you're looking at tumors, the virus may be in a particular form, or it may be basically expressing itself, so it will look just like an mRNA from the cell, in which case you have to sequence much deeper because you also have to get over the host messenger RNA. For example, the group that discovered Merkel cell polyoma basically found one sequence out of several million that looked like a polyoma, and then right away they knew. I mean, you can tell when you have a very significant alignment that there's a new virus. So that's the path that we're on right now, looking for viruses associated with acute flaccid paralysis, and we found in those.
I'm sorry, what's acute flaccid paralysis?
Is the disease caused by polio, and polio being now on the verge of eradication. There's a lot of attention paid to this symptom, so that when kids come down with acute flaccid paralysis in countries where polio is still present, they will be immediately flagged. The WHO collaborating groups will be collecting stool sample from the kids, testing it for polio. And if they find polio, they establish a ring of protection around that child by heavily vaccinating the families and the whole neighborhood, basically.
It comes from smallpox eradication, the ring strategy of
That's right.
identifying cases and then,
But actually, most of those viruses we found, we found from stool, from kids from Pakistan and some from Nigeria, who had this disease, but did not have polio. Something was making them sick. And we found several viruses in those samples. We're still not sure that any of them are responsible for the symptoms, but that's the origin. Kids tend to get a lot of infections, tend to have higher viremia. And we were very fortunate to have collaborators in Islamabad who sent us basically their stool samples that they'd kept after looking for polio. Dr. Zaidi was the group leader in Pakistan.
Do you find any bacteriophages in these stool samples?
Of course. No, we have a huge database of bacteriophage sequence. We're focused on eukaryotic viruses.
Yeah.
Sort of a bias from my origins as a retrovirologist. We'd love to find collaborators who could crunch this data. There's also viruses we find frequently in blood samples that are harmless, or at least thought to be harmless. There's a whole family of very diverse viruses called anelloviruses, also called sometimes TTV.
Like circoviruses,
yeah,
right?
they're actually phylogenetically a bit distinct, but like 100% of the people have those viruses. You probably acquired them either at birth from your mother or very soon thereafter from siblings and other kids. So we always find those, and we have tons of sequence data. It's very diverse. This is probably the virus that's been co-evolving with humans for the longest time, and it's 100% prevalence if you look hard enough. 3 species, hundreds of genotypes. For all we know, it's a harmless commensal virus that's evolved to become at one with humans.
So this, I'm trying to remember. I'm looking it up on Viral Zone here, which I've come to use all the time.
It's a great site. They're probably listening.
Single-stranded DNA, circular DNA viruses, is that
Anello.
Right? Small, right?
Very small genomes. And these are the type of virus that you found in the rotavirus vaccines.
Well, so these were part of the circovirus family. They're very closely related. They probably originate, if you go back deep enough, as plasmids. They have the features of plasmids about the same size as smaller bacterial plasmids. But they're definitely eukaryotic viruses. And, the whole vaccine incident, if I may call that, having this technology to find viruses. I was reading this book by Arthur Allen about vaccines, sort of a nice historical review of vaccines going back, way back with smallpox. And it sort of inspired me to look at what else is in vaccines beside the expected attenuated strains. And we found in the Rotavirus vaccines a circovirus. That is a pig virus. The strain we found was not pathogenic in pigs, but it is a very common pig infection. And of course, I was concerned that this would be used by people who are against vaccines in general as, look, we told you so. So, we alerted the company, and they very rapidly, confirmed that, got in touch with the FDA. The FDA had to, I think, follow the book, which is, there should not be anything but the virus that you claim is there. And it's unfortunate because this Rotavirus vaccine, like the Rotatech vaccine from another company, are so effective that in the U.S. alone, they've reduced the rate of hospitalization from diarrhea by half in just a few years. These are really recent vaccines. They're oral vaccines, so you don't have to poke your kid with a needle. And they're extremely effective. And especially in the developing countries, these vaccines will reduce mortality probably from diarrhea probably by half. Because they protect against rotavirus, which is one of the major, child killer in developing countries. So luckily there was two vaccines. The FDA sort of clamped down on one, but I've since basically realized that the circovirus is not a pathogen. It doesn't even infect humans, and it's like a harmless extra piece of DNA in your oral vaccine.
So what's the current thinking on how it got there?
People suspect that it came from trypsin, which is derived from pig pancreas, that is used to actually activate rotavirus. You have to digest its outer shell to make it fusogenic. And also when you passage cells, you typically use trypsin to disaggregate them and transfer them to a new plate. And it's quite likely that this pig virus came from this pig trypsin and became established. And as a matter of fact, what was reassuring is the this pig virus was present in the vaccines from the very beginning, which means that all the clinical trials were done in the presence of this virus, and they showed no evidence of any problem. The vaccine's very effective, both vaccines, and so all this information reassured the FDA, and they basically allowed the company to continue to sell their vaccine, and with the promise from the company that yes, we're going to go back and clean it up and remove this contaminant.
No, so they're going to remove it.
They are.
We understand
it's going to cost them some bundle.
Yeah.
And turns out that the other vaccine was also contaminated with PCV1 and a relative PCV2 at much lower level. But, the same conclusion. It's not even infectious for humans. So
I have to ask a question here. This is a little complicated, but it turns out that I serve on a committee that is a subgroup of an outfit called the Brighton Collaboration. I don't know if you're familiar with these guys. Brighton Collaboration. This is a group that was established in 1999 by a group of individuals. Notable amongst them is a guy named Robert Chen, who for 15 years was head of a vaccine safety group at CDC, and more recently is in charge of the HIV vaccine trials part of CDC. This is an international group that started kind of, I guess, ad hoc, except that Bob was working for CDC at the time, and now has input from the European CDC and the American CDC and the World Health Organization that has the goal of basically on an international level examining vaccine safety and trying to come up with international standards and guidelines. They are not a regulatory outfit, okay? But they're trying to come up with international standards and guidelines so that everybody can be kind of on the same page with vaccine safety. The subgroup that I work on deals with vaccine vectors. So at any rate, we meet by a conference call once a month. And last time we called, this PCV thing came up, okay? And two of the individuals on the conference call were talking, apparently had some conversation outside of the conference call, and were talking about this issue and wondering whether they were thinking that the primary thing that was, they thought the discovery technique was primarily microarray, and they were talking about whether it would be useful to use microarray as routine screening of vaccines. They decided, well, no, because you got to know what you're looking for. Okay, you only find what you're looking for. And I spoke up because of my experience with TWiV and having looked at your paper on TWiV and said, well, no, you could do deep sequencing and maybe be able to even identify, lots of things that you don't know what you're looking for.
That you didn't know existed.
So they said, oh, good idea, Condit. Okay, why don't you investigate this? So here I am. I'm talking to the man. I'm investigating this. Is this a practical thing to do? Like screen vaccines for adventitious agents by deep sequencing?
I think especially if you do it early in the process of developing your vaccine, you prevent nasty surprise down the road where the FDA is going to stop your vaccine and it's going to cost you a bundle. It's relatively simple and it's not that expensive. And I know there are companies now providing this service to vaccine developer. We're going to sequence your cell line, make sure there's no infection that you didn't expect. We're going to sequence your viral seed stocks to make sure there's nothing unexpected. Now, you can sequence or you can microarray. I mean, microarray is a great technique. It's probably a little bit cheaper. It's certainly a bit faster. But it does limit you to only recognizing viruses that are closely related to what you already know. So if a virus is too different, it will not stick to your microarray. On the other hand, since by the way we do our metagenomic, us and many other groups, is we generate the DNA sequences, then we translate it every 6-way from both strands, starting every 3 nucleotides. And then we take that sequence, that protein, sort of virtual protein sequence, and then we compare that to viral protein sequences. That allows us to see a little bit further than microarray, where we can recognize protein-level homology, while the nucleotide-level homology has disappeared. It's evolved too far from what you already do know. So there's room for both. I think microarrays are probably a little bit more established as a technique, a little more routine, maybe a little more sensitive to low copy viruses.
So a couple of questions then come up. One is this something we should be doing? And the other is, when you find, what do you do with the information? Because you're going to find all kinds of stuff, right?
Well, if you find a virus early, you junk your cell line that got contaminated along the way, and you try to find a cleaner cell line.
Okay.
So in the case of the porcine circovirus, if it's in trypsin, what do you do?
So how do you prevent reintroduction?
Yeah.
So it is ubiquitous in pigs. And so why don't you get trypsin or some protease enzyme from somewhere else? I think it's tradition, basically. People have been using, pig trypsin for years. You can buy recombinant trypsin. That's going to be very clean, but I understand that's a little more expensive. Or what people actually do is they irradiate their trypsin to kill any viruses that might come along. But apparently it was not sufficient to kill these circoviruses. I mean, some viruses are quite resistant to irradiation.
Oh, these guys are really small.
They're very tiny.
Target size is a factor, right?
It's like less than 2 kb genomes. They're very, very tiny.
I think they're more resistant than prions to the radiation.
That's pretty resistant.
I mean, of all the viruses, all the viruses that Stan Prusiner tested, and he published a nice graph of this inactivation of viruses of different size genomes, DNA, RNA, with ionizing radiation. Prions were only slightly less resistant than circoviruses.
Really?
Amazing. So that's probably why they're...
And I know prions, you can autoclave these things. They're still
maybe that's something
active.
else, yeah.
So you don't want to autoclave your trypsin. So I think now the FDA is going to require some testing of new vaccines. Now, vaccines that are already out there that are being used, if the virus that's contaminating them is not a pathogen, I think they're going to let it slide, or at least give the company time to clean up their vaccines.
I would guess they're all going to have to be cleaned up, because the public, being aware of immunization now,
the public
they're not going to
pays a lot of attention to this. Yeah. But we looked at most of the live attenuated vaccines, and we only found one that was contaminated. Right. Now, we could have gone deeper, and clearly we missed PCV 1 and 2 in the Rodent. Yeah. We didn't go deep enough, or it's a lot-to-lot issue.
So just to give a little historical perspective on this is good, because I just came from a poxvirus meeting, and we have this meeting every 2 years or something like that. Now, the smallpox vaccine is arguably not, by today's standards, not the safest vaccine, but certainly the most successful vaccine that we've ever had, okay? And the smallpox vaccine that was used during a vaccine eradication campaign worldwide was made by standards that would be totally unacceptable today, where you grew it on the bellies of cows and harvested it and this kind of stuff. And we were having a conversation about this. You can take this and streak it out. That's microbiology talk for take some of this fluid and put it on an agar petri dish. Okay, some of the old vaccine, all kinds of bacteria grow out of it. Okay, and I'm sure that if you did actually you ought to do that. It's just out of energy. Get some dry vax.
I don't know that smallpox vaccine is available anymore.
Oh, you can get dry vax. Okay. If you can't, I'll get it for you. Okay, it's around.
Yeah,
you find all kinds of stuff.
I mean, the new vaccine made by Nordisk, is that the company in
Bavarian Nordic?
Is that an infectious vaccine?
Yes.
Yes. So that is being stockpiled for bioterrorism.
Of course, yeah.
You should look at
that.
Now
that's a cell culture vaccine.
Yeah,
that would be,
who knows, right?
Who knows what's in it?
So unfortunately, the postdoc Joe Victoria, who did this really nice vaccine study, and he's moved on. He's now working for Boehringer Ingelheim. He's hunting for
I'll come and do a postdoc with you and do
viruses.
it. Okay.
Okay, your sabbatical in San Francisco.
Great. Yeah. Right, can you imagine? I could learn this stuff and bring it back to my lab, right?
Sure.
And then sequence the forests.
That's right. You got a new career, Vincent.
Yeah. No, it's great because we get people saying, can we come over for a month or two and we'll bring our sample. So I try to accommodate visitors. We had people from England looking for encephalitis-inducing viruses. Unfortunately, we didn't see anything for that case. But they're freezer fulls of interesting clinical samples throughout the world, just waiting for a bit of deep sequencing.
You said, so all the data that you generate, what happens to it? Can anyone see it, or do you keep it?
Well, luckily we have a great collaborator, Chunling Wang at Stanford, who has established sort of a small computer cluster through which we push our millions of sequence reads to look for sequences that look like viruses at the protein or nucleotide level. And he keeps that organized. There's no way you can have notebooks with that sort of information.
So the data management must be just phenomenal.
It is. And then the eyeball time, I mean, when we get the data back from a 454 run with a million read and all the analysis that go with that, somebody has to look at that. Now, luckily,
wait a minute. You just said eyeball time. Is that right?
Well, yeah.
It's a new term for me. I like that.
Where you basically sit in front of a computer and you scroll through the most interesting hits. And we have a way now to bring the most interesting hits to the top.
So hits mean you've taken all the sequences and asked, is there anything known that they match up with, right?
Correct.
That's a hit.
Yes.
Okay. Sorry.
And if it looks like, we're finding a lot of small, mostly because we're looking at a lot of stool samples, we're finding a lot of small genome, RNA genomes in the Picornaviridae family, astroviruses, circoviruses, the kind of enteric viruses.
Right.
Blood is much more sterile. Obviously, and we do find a lot of bacteriophage in blood.
Why would it obviously be more sterile? Because it comes from people, right?
Well, because in a sense, the most contact you have with the environment is through the food that you eat. I mean, sure, there are viruses that infect your skin, that can through injection equipment, through sex. But in a sense, your contact with the outside world is really through your gut. And therefore, there's a lot of enteric viruses that I think remain to be discovered. We've looked at blood, working the blood bank. It is another focus of ours. But we find these common anelloviruses, this flavivirus called GBV, which is again quite a common but probably harmless infection, and a lot of bacterial DNA. And bacteriophages. And so either this is leaking from the gut, where we know there's a lot of bacteria, and therefore bacteriophages, or they're actually small foci of bacterial infection in humans that we didn't know about. We're not, equipped to follow up on these bacterial and phage hits, but we do have a fair amount of database.
So when you find these in blood, is that blood not used, or is it not a problem to use it?
No. Blood right now is, if there's any trace of HIV, hepatitis B, or hepatitis C, it is thrashed. The people are contacted, they're counseled for treatments, but we don't want their blood for transfusion.
What about when you discover a different virus in blood?
So these anelloviruses and these flaviviruses are so prevalent that if you were to exclude people with that, you'd have no more blood. And because they're not pathogenic, as far as we know, and because blood transfusions are crucial. I mean, you're in a car crash, you're bleeding, or you're on an operating table, you need the blood. And because there's no evidence whatsoever that these are pathogenic, it's basically tolerated in the blood supply. Blood is a very complex mixture. There's companies have always tried to make artificial blood, but it is, in a sense, very safe and very cheap to just bleed out volunteers and then test that and transfuse it.
But it gets more as time goes on, and we know more and we can detect more. The whole maintaining the blood supply becomes much more complex.
Oh, because now the blood supply is being tested not only for these viruses, but in certain regions at certain times for West Nile virus, because that can be transmitted through blood transfusion. So whenever there is certain seasons of the year when mosquitoes are out, West Nile virus RNA testing kicks in, and every time you add another test, the price of blood goes up, and people are always trying to limit, the cost of healthcare, and so you can't test against you should overreact to every virus, whether it's a circovirus that doesn't infect human in a human live attenuated vaccines or another virus in the blood supply. You just wouldn't move. I mean, every time we open our mouth, we'll probably inhale a few viruses.
Well, that's one of the reasons I wanted to bring up the old smallpox vaccine story, because, you know, by today's standards, that vaccine would just be a nightmare. But at the same time, it was the most successful vaccine responsible for eradicating a terrible disease.
Live attenuated vaccines are extremely effective. They're cheap to make. Now, there's been some accidents. I mean, polio occasionally reverts. You know, there was some hepatitis B that were contaminated back after World War II because they used to add, for some reason, some human serum. And so hepatitis B was contaminating, I forgot another vaccine, but there were huge outbreaks of hepatitis among recipients. That was the yellow fever vaccine that was contaminated.
Right, I remember that. Yeah. So is there no reason to look at inactivated vaccines then?
Well, we chose, Joe, Victoria, and I decided to look at live attenuated because if there was a contamination with another virus in an inactivated vaccines, it would be killed along with the vaccine virus.
Yeah.
So it would be much less of a concern. So we focus on live attenuated vaccines. But I think eventually people will look at, inactivated ones as well.
So you said something during lunch that I want you to tell everyone. You said, I see DNA all around me. There are...
Well, I see viruses.
Viruses everywhere.
You see viruses everywhere?
Well, no, my eyesight's not that good, but I've been trying for many months. I thought I just want to go to the dog run and just start, collecting little blue bags and see, what's infecting dogs. I want to look at bird guano from, pigeons and, whatnot. I mean, there's a universe of viruses waiting to be discovered, and these techniques are really allowing us to do that. Now, of course, as a virologist, most virologists don't spend the entire career on one virus, one real nasty one. So polio, HIV, pox viruses. But now we can work on many viruses. For me, I've had to learn all about viruses I knew nothing about. So the big taxonomy book is sort of our Bible now.
Do you do experiments on any of the viruses you discover, or is that not possible for you to do?
If we had the time and the funding, I would love to grow those in tissue cultures and develop antibody tests and express their surface antigens and find out how common, people seroconverts, where, at what age. I mean, all the biology, basically. But we've been so, lucky and happy just sequencing these things. And then we do quick PCR tests to find out how common they are in certain populations, that we've stuck with the nucleic acid work pretty much at this point. Turns out once you publish a paper about a virus and people have the sequence, they will either go and fish it out themselves and develop this serological assay. I mean, it takes a life of its own, basically. People,
right, so you inspire a lot of research that other people...
Yeah, and, there are half a dozen groups that do nothing but viral discovery. And every time they, other groups then kick in or people, or groups with more interest in protein expression. The day we start finding no viruses will slow down and do the biology.
That's a good question. When will that day come? I mean, assuming we can continue efforts like yours to search, when will the day come when we've found them all or most of them?
Turns out that in human, we've noticed that now in human stool especially, several groups are finding the same viruses within months of each other. So we may be hitting sort of diminishing returns. But my feeling is, as you start looking in different parts of the world, going maybe to blood as well, there's still some low-hanging fruits for humans. Now, when you start thinking about animals, then it's wide open. I mean, there's all the species of mammals to look at.
And plants.
And plants.
Insects, fish, sea mammals.
Insects.
I mean, basically, we can't do it all. There are too many viruses for us to ever discover all of them.
I mean,
right? Yeah. But I think, from my viewpoint, it is absolutely fascinating and essential to learn about as many as we can. And as we said before, all this dark matter, all the viruses we don't know about, certainly understanding them will help us understand evolution of not just viruses, but living things.
And also in terms of diagnostic, as we characterize more human virus, let's say, people who design microarrays will include these new viruses on the microarray. And I think there's a good future for that in the sort of clinical medical world, for the rapid testing of your sample. Maybe some few years from now, you'll go to a doctor's office, and 10 minutes later, you'll know every viruses you have, in your lungs or elsewhere.
Have a better scenario.
Again, finding a virus doesn't mean that it's making you sick, or it's ever a pathogen. But these are sort of, downstream questions. First, you get the genome, and then you can ask questions about the biology.
I have the mirror scenario.
Yeah, that's right.
I think someday you'll stand in front of a mirror in your home and it'll tell you whatever infections you have, or you're clear for today. Have a nice day. It's going to do that.
I'm convinced we are undergoing, especially children, frequent infections, especially with the enterovirus sort of family. I mean, there have been a few studies when people look at stool taken weekly from kids in daycare, and pretty much, every week or two, they will find a virus.
Sure.
Now, kids get sick a lot, but not that often. So most of these go right through, maybe a little sniffle or whatnot. But and for all we know, it's actually good for the immune system to be teased like this, to be sort of educated. And, the whole hygiene hypothesis is quite interesting, that if your immune system sees a lot of harmless or not very pathogenic viruses as you grow up, if you get hit at some point with a very nasty relative of these, like a poliovirus, if you haven't seen the relatives before, the poliovirus will really hit you hard. But if you've been educated immunologically by these prior infections, you might feel nothing.
Yeah, this concept really fascinates me, because my sense is that we're overdoing the hygiene bit. Everybody's carrying around a thing of hand sanitizer with them now. And, there's a certain amount of that that's okay, but the hygiene hypothesis suggests that you could overdo that.
And it's bad for your skin.
So I found just yesterday, CDC made an announcement with respect to rotavirus vaccine. They just indicated that they should not be given to babies with severe combined immunodeficiency.
I think that's always been the case.
They just announced yesterday in Morbidity and Mortality that it's another contraindication.
Is that just the rotavirus or any
Just
infection?
rotavirus in this specific announcement. But I wanted to ask you if that is a good idea, because maybe the circovirus could be a problem in SCID infants.
It's always tough to put any infectious agent in an immunodeficient host. But they can't live in a bubble either. Well, sometimes they have to.
Yeah, because in immunocompromised people, normally benign viruses can cause a lot of problems. Right.
But in the case of the circovirus, I think the concern there is the rotavirus live attenuated strain itself, not the circovirus, because that's not going to infect anybody, any human anyway. It'll infect a pig, but not a human.
I just want to go back to this I see viruses everywhere thing. We had Eric gave us a tour of the city today, so we were driving over here from the hotel, and ordinarily tourists, people would point out buildings to you or landmarks of one sort or another. And Eric's saying, "Oh, that's a great spot for a collection. Oh, we can get these kind of viruses over here."
Yeah, that was great.
Well, there are neighborhoods in San Francisco with a lot of hepatitis C transmission, a lot of HIV transmissions, with different risk groups, basically. Right. And they're quite concentrated. Yeah. And there are many studies in the city where people enroll young injection drug users or gay guys and then basically they'll donate samples on a frequent basis, which are then put through a whole lot of tests. It's a great HIV and Hep C epidemiology town.
So how's your laboratory funded?
We are lucky enough to get some hard money from blood systems. Okay. I also, like most people, read NIH grants. Okay. So right now I have an expiring R01 grant that I need to renew. And we also have collaborations with people that will make us co-PI on their grants. I also do dabble in sequencing, common pathogens like HIV, Hep C, and Hep B. So that brings in some funding.
Okay.
Well, maybe we'll see his grant. Who knows? It's great stuff, Eric. Love it. I love discovery. Like most people, Christopher Columbus discovered because he had to.
Right.
He loved it. We're all interested in discovery. You and I have worked on a virus all our life. We do discovery, but I think this is fabulous.
Right.
We do have to put up with people saying, this is a fishing expedition. You're just collecting stamps.
Not from us.
No, you won't hear that from us.
It's not. It's clear, and that's one of the reasons why we wanted to have you here, because maybe a lot of people do think it's stamp collecting, but it has far more implications than that. And I think it's essential that we find out the whole virome.
Right. I think the technology, there are phases, I think in virology, and one right now, just massive sequencing provides very interesting starting point for the biology, the tropism, the pathogenicity. But first, the genome. I think the genome first really helps.
Need to keep doing this until the dark matter is no longer dark.
I think that's an endless pit, if I could say, of information.
It's asymptotic.
And it's also always evolving. I mean, viruses keep coming in from animals. So if you sample a population, over decades, you will see changes.
Let's go ahead and do some email. Normally on TWiV we do some emails. I have a couple that are sort of related to you, and so we'll do that. But before we do, I want to acknowledge the support of this episode of TWiV by Data Robotics, the makers of Drobo. Eric, you have any Drobos in your lab?
I don't know. I have a little Mac attachment thing that sort of a generic Drobo.
You put all your data in the cloud, right? Basically.
Well, our sequence information actually is not on my computer. It's on some hard drive at Stanford, which is backed up on a regular basis.
Well, Drobo's great for labs that generate a lot of data like yours. They are storage solutions, multi-drive enclosures where the data is redundantly stored. And they have 4, 5, and 8 drive models, which you simply plug into your computer, and they transparently act as backups and redundant backups. So if any drive fails, you just pull it out and put a new one in. And as I say, they make many different models. You might want to start with a 4 or 5 drive model, as I did. I started with 1. I now have 3. They're terrific, and they make a great product. They're so neat. And the latest one that they have is a file sharing model. It's called the Drobo FS, which you can hook up to a network, and up to 16 users can share it at the same time. It's really beautiful. Now, we just finished the 3rd Drobo giveaway contest. And the winner is Eric in Birmingham, Alabama. I wonder if Eric's a scientist. Who knows? We'll find out.
We'll find out.
Congratulations, Eric. So the 3 giveaways are over. But you can still get 50% off the price of a Drobo S or a Drobo FS at drobostore.com. All you have to do is fill out the questionnaire at bit.ly/TWiVDrobo2, all one word. It's the same questionnaire as previously, but Drobo would like to have a little more information about who's listening to podcasts about science. So if you just go there and fill out the questionnaire, you get half off a brand new Drobo.
That's a good deal.
So that's a good deal. And check that out. And as usual, we thank Drobo for their support of This Week in Virology. Let's do a couple of emails. First one is from Stan. I found your podcast through the Microbe World website. Thanks to Microbe World, I've been unable to sate my thirst for knowledge about the microscopic organisms that act as the foundation for our planet. So I stumbled across a podcast about viruses. Hell yeah. Until recently, my RSS feeds have only been filled with skeptical podcasts, making me the residential expert on why homeopathy is bunkum, but now I get to amaze my roommates with fascinating info on viruses and their ilk. That's good. When I first discovered your podcast, I jumped headfirst into the latest episode and was kind of lost, almost bored. Not one to give up so easily, I decided to take it slow and go through your previous episodes. Clearly, this was the best strategy because your first episode on West Nile was so jam-packed with information on a virus that I knew almost nothing about. Ever since, I've been hooked. In a world where ignorance is praised and brainless celebrities reign over cable television, people like yourselves, whose only agenda is to educate, give me hope for the human race. Wow, that's pretty serious
very
stuff.
nice. That's very nice.
In summary, I love the show and wish you best of luck in the future. Of course, my best wishes are somewhat selfish, as I hope for more episodes every week. Well, thank you for that, Stan.
We'll do our best.
Then we have an email from Darnell. I am studying virology at Columbia University and would like to pose a question to the expert panel of virologists at TWiV.
Is this one of your students' questions?
I believe it is one of my students. I asked as an extra credit assignment for some questions, and here's one. In the most recent lecture, we were introduced to vaccines, and my question pertains to such. It is my understanding that the same virus that causes chickenpox that we get when we are younger also causes zoster rash, yet 2 different vaccines are given to treat these diseases. Why is this the case? Are the degrees of the vaccines different given that they are delivered to different age groups? Why doesn't the immune response developed after chickenpox protect you against zoster rash?
So my understanding is that the zoster vaccine is in fact the same as the chickenpox vaccine, but a higher dose. And the reason they give it to old guys like me is because when you get old, your immune system gets tired and it's time for a boost. Basically, it's a booster, okay? That's why you get shingles when you're older, is because your immune system, this is hard to talk about. Your immune system starts to crash. Well, not crash, but at least get a little tired.
We'll call it waning. How's that?
Okay, waning. I can do that. And so it's the same vaccine in a higher dose, and you're basically getting a booster.
Yeah, that's it. Sheldon writes, this is for you, Eric, the likely culprit for the PCV contamination of the rotavirus vaccines is trypsin. I came across this statement in a document that described the manufacturing process for Rotatech. Rotatech manufacturing process, the applicant has put adequate measures in place to ensure absence of adventitious agents for the above materials, and the virus removal or inactivation steps have been validated. So he sends us a document which apparently has information on the production. And he asks, does the gamma irradiation kill the PCV viruses in the trypsin? The document I referred to had trypsin irradiated before use in production. Could the problem be that the trypsin used at the very start of the process was not irradiated? I really enjoy listening to your podcast.
In a sense, it's quite possible that the irradiation prevented superinfections with circoviruses, but that the virus was there from the beginning. I mean, the cell lines that people used, the Vero cell line in that case from an African green monkey, has been passage for probably who knows how long. Yeah. So when it gets to the company, it's already gone through multiple hands, and it's quite likely that not everybody was so careful.
I guess the key is, as we said, to start with a cell line that is clean, that you determine to be clean by sequencing, and then just keep it clean.
And then you're going to make sure, yeah.
Patty writes, Dear Professors, I've been a loyal listener since 2008 and sent an email around TWiV 15 asking for clarification on definition of infectious and contagious. At that time, I had just enrolled in a virology course after work, and now, having completed several of the courses, I am now enrolled in the Master of Science Virology program. At 43 years and with a couple of kids, I did a great deal of thinking about this, but I am thoroughly enjoying it and am occasionally mesmerized by the beauty and complexity of viruses, as well as awed by the devastation they can cause.
Gotcha. We got him.
It's good. We hooked another one. Isn't that great? And he sent us his last assignment for his virology course, and he sends it to us as a... he dedicates it to us for helping him get through the course so far. And that's pretty cool. I enjoyed that a lot. One problem that I have encountered is that getting my hands on pertinent recent publications that explain concepts at a level that would be completely useful to me is, at this stage, quite hard. The publications in PubMed are very often a bit hard to decipher and very focused on specific issues. I understand why this is so, but finding references for assignments is quite hard. But I am sure that it gets easier over time, especially when listening to TWiV. You got any advice for him?
You know what? Wikipedia.
Wikipedia helps, yeah.
I use Wikipedia all the time, and it's very readable, and I was actually going to use that as a pick one time. I'm a little hesitant of this because it is, done on the fly. You do worry about whether or not it's going to be entirely accurate, but I'll tell you, from most of the stuff that I read seems accurate.
I think you can Google things, you can wiki it, but keep listening to TWiV.
Yeah.
Because eventually,
it gets easier.
It gets easier. If you listen, you don't have to listen every week, but if you listen enough, you're going to get the feel for all this jargon and language and, the way things work.
Starts to you build a framework, so that the new information sticks on the framework. It does get easier.
So here's a question now, which Eric can help us with. What are the steps to uncover a virus that has not been previously identified? I have enclosed a brief description of one such suspected virus known as RRV, or Rodent Respiratory Virus. I have thought about looking a bit closer at this because it has some relation to my current job. I have enclosed the text sheet from this. As yet unknown virus. Insofar as the pulmonary lesions are concerned, it would look from the pathology that it could seem to be something like the RSV subfamily of paramyxoviridae that human neonates can sometimes get. I understand the principles of PCR and multiplex PCR, but I can't see how you identify something entirely new with them. Can you at some point discuss this, and perhaps also how they have uncovered some in the past? So I think we talked a little bit about this today, but how would you answer that?
Well, if you suspect it's part of a certain viral family, like related to RSV, you can take all the RSV in the databases, or all the members of that group of viruses, and find a conserved region among them, to what's called a multiple sequence alignment. You can then basically design primers that will amplify pretty much all the paramyxoviruses out there. And then you can rapidly, PCR takes just a few hours. You can buy primers for, $20. You will know whether you have a paramyxovirus. It's a bit harder to do for some viral family that are extremely diverse, but for some other, like let's say the astroviruses, you can do some consensus PCR approach quite easily. You can also use microarrays if you have a group you can collaborate with, or make them yourself. Or do the deep sequencing approach, this metagenomic. And again, if you have something that grows in tissue culture, as what it sounds like, you have so much virus there that you really don't need to go very deep. You can sequence, if you can subclone, you could do that basically pretty much in your kitchen for a few $100s.
So I just had a quick look at the text sheet that he sent you. He works for some sort of animal supply company or something like that, and they've got a pathology in some of these animals, right? So he can just rip out a lung and send it to you, right?
Yes, we prefer fluids than tissues, but it also works with tissues.
And Patty, we've talked in this TWiV about how to deal with the dark matter, the stuff you don't recognize. You just try and find motifs maybe that point you in the right direction, but it is very hard.
I didn't get to ask this before. Currently, how much does a deep sequencing run cost?
There's all kinds of different depth. A 454 run, which will generate about 1 million reads of about 300-400 base pair sequences, will cost anything from $10,000 to $25,000, depending on what company is providing the service.
But you could do multiple samples in that.
Yes, so you can do more than just one. You can use tags at the end of your so-called random PCR primers, so that you can then recognize which of those 50 samples that sequence came from.
I would imagine that this is something that's going to get a lot cheaper quickly.
Yes, it already is. I mean, it's dropping. The throughput is just keeps leaping, and people are talking about third generation sequencing, which will be even cheaper. I mean, people are shooting for the human genome for, was it $1,000?
It's amazing. $1,000 genome. Have you sequenced your genome yet?
Not yet.
You plan to?
I think I would, yeah.
Might as well. Sure. Look at your polymorphisms, your risks.
There's some things you'd rather not know, but I think I'd be curious. I mean, the problem is the analysis. What do you do? I mean, everybody has some kind of defective genes or not optimum genes in their genome, and you don't want to overreact to that.
Yeah. Okay, we have just a couple more. One is from Joe. While I was listening to your discussion of the antibodies we have to the alpha-1,3 sugar group and how it could be used to target disease agents, I had a thought. It struck me that it would seem to be very much easier to use this as the ultimate bioweapon and target a key functional group in our bodies than try to target HIV, which is always mutating. As this is not my area, I hope I am wrong. It seems that one could target a surface feature on heart cells or lung cells or liver cells and cause the body to attack itself in a way similar to the cytokine storm discussed related to 1918 flu. As I said, I hope there is a technical reason that makes this impractical. If not, please do not read this on air, as I don't want any knuckleheads to get any ideas. So Joe is an environmental health and safety manager somewhere, and I can understand why he might be concerned. So this is based on the idea that we don't have alpha-1,3 linked sugars on our tissues, but the bacteria in our gut have them, and so we make a lot of antibodies from the day we're born against these sugars. So any pathogen that infects us that have such groups are immediately destroyed. It's a very early form of the innate response. And I think we were discussing Kerry Mullis's idea where he would hook these up to aptamers that would target them to specific viruses like HIV or flu. So there's no problem with our tissues because we don't have the alpha-1,3 sugar group, basically, right? Did I get it right?
Right.
Thanks, Joe. Elena writes, "Hey guys, I have a job assisting a medical professor in doing literature searches and compiling information for the new edition of a book he's writing. The other day he asked me to gather some information on the XMRV virus. Having listened to episode of TWiV you did on XMRV a while back, I was able to converse knowledgeably with him about it right away. So I guess I just wanted to say thanks for helping me impress my boss. Virally yours, Elena." I like that.
Yeah, that's very good.
And our last one is from Welkin. This is about endogenous retroviruses, which I know Eric likes very much. Dear Vince and Dick and Rich Condit, congratulations on producing a brilliant series of podcasts. I have been hearing about TWiV almost since you began podcasting, to the point where I had come to believe I was a fan without having actually listened to a single episode. I even deluded myself to the extent of recommending it to others. With the recent purchase of an iPhone, I have made up for this fraud, and I'm working my way back through all the episodes. I especially enjoyed the episode on reverse transcriptase and the discussion on endogenous retroviruses. HERVs were the topic of my thesis when I was a graduate student in John Coffin's lab. Dick was amazed that we humans haven't gotten rid of all those useless retroelements from our genomic DNA. In point of fact, we have jettisoned them by the bucketful over the course of evolutionary time. The sheer abundance of these elements is all the more amazing then once you realize that the ones we know about are just a tiny fraction of the whole. These are the pernicious few that made it through the evolutionary grinder and still cling tenaciously to our genomes. It's quite the poet, isn't he?
Welkin writes very well.
The most obvious evidence for loss comes in the form of solitary LTRs, the element left behind when recombination between the two identical ends of the ERV loops out the intervening retroviral genes. Solo LTRs vastly outnumber intact proviruses in our genomes, indicating that large chunks of them are more frequently lost than not. There are other mechanisms of loss, and probably a majority of integrations that land in germline DNA simply don't survive down the generations. So we don't know about all the stuff that was there once upon a time. It is staggering to realize that retroviral epidemics like the current HIV epidemic have a constant presence throughout evolution. Keep up the great work, and if you get a chance, check out Small Things Considered, Elio Schechter's blog on microbes and viruses. Yours truly is an associate blogger. Thanks again for all the great listening. So actually, Small Things Considered was a pick a long time ago on TWiV. It's a great blog, and thanks for writing, Welkin. Sounds like we ought to get Welkin on sometime and talk about herbs.
Absolutely.
All right, that'll do it for email. Let's do a couple of science picks this week. Eric, you know, every week we pick something, and if you have something that comes to mind, feel free to chime in. Rich, what have you got?
First of all, we ought to put in the show notes links to the Brighton collaboration, okay, so that people can check that out.
You're going to send me those. Oh, you already did, yes.
Well,
if not, I'll get it.
We'll figure it out. And they're updating their website, so keep an eye on that. So my pick is something I was thinking about the oil spill the other day, and I was wondering, you know, what's the latest, where is it, that kind of stuff. So I looked on the web to see if I could find a map, and sure enough, there's a Google site that tracks the spill. and I've got the web link in here, but even the name of the web link is relevant. It's www.google.com/crisis-response/oil spill. And Google has done their usual thing. They've hooked this up so that they can basically collect data from all over the place and stick it on a Google map and show you the history of the oil spill, the predictions of where it's going to go, where it's at today, the impact on the environment, and all sorts of other stuff. And you can click on little boxes that give you different overlays as to what the impact of the spill is. So I found that unfortunately interesting, because this is a big deal. You know?
It is a big deal. Yeah, it's something everybody's aware of and everybody worries about, but not a lot of solutions so far. Nope. Well, my pick is a pick suggested by Jim, a longtime listener from Virginia, and he suggested a page at the Howard Hughes Medical Institute. It's called Resources for Teachers and Students. And I can't reach it right now, so I can't tell you much about it, but it's a great page which has exactly what it tells you, a lot of video resources, a lot of imagery, all having to do, of course, with science and science teaching. So if you're teaching science, great resources for you in terms of presentations and explanations, videos, PowerPoints, and students will love it as well.
Here it is.
Exploring well, that's one of the that's one of the featured things they have. It's called the 2009 Holiday Lectures on Science, Bonnie Bassler and Baldomero Oliveira. So it's great stuff, and thanks for that, Jim. Eric, you want to recommend something?
I love the history of science, and especially microbiology books. I think it really helps to get the bigger picture, sort of historical context. And the vaccine work we did was actually inspired by this book called Vaccine, the Controversial History of Medicine's Greatest Lifesaver. Something close to that, by Arthur Allen.
Arthur Allen.
And it describes the very deep history of vaccines, starting with the smallpox. And I think this show was mentioning how in the old days smallpox was just so prevalent. People were willing to take incredible risks to protect themselves. They would literally scrape off pustules of virus and shoot them up their nose. Extremely dangerous. But if you didn't kill you, it made you stronger. And the people did the cost-benefit analysis, and they went for it, because the alternative was to die of smallpox in this very common epidemics. And it's a great book, and it goes through the history of vaccines, the good and the ugly, and how safe they are now, the great benefit they've brought to mankind. And when I stumbled onto this contamination in Rotavirus, I was really worried that it would be spun the wrong way, and luckily I think the companies and the FDA have really behaved as is their role. They did not overreact.
So this book is presaged your investigation of the vaccine?
Yes,
right?
This was really I read that over a vacation. I thought, there's all this sort of precedent. And we all know about SV40 and polio reversion. I mean, there have been accidents.
I think it's great when you have the source of inspiration for a scientific experiment. So.
And this is, and the work you've done is, I think, is going to have a tremendous impact, because it's just as in the story that I told with the Brighton collaboration, it's raised consciousness to another level that we need to keep in mind in looking at vaccines.
And there is an initial shock to it, but I think eventually it will prevent accidents.
Right.
It will
Absolutely.
reassure people that, look, everything is being done to make these things as safe as possible. Right. And they are very effective.
Great pick. If you're a new listener to TWiV, please consider subscribing in iTunes. It's free and allows you to automatically receive new episodes as they're posted. If you don't use iTunes, you can always come to twiv.tv and listen, as well as look at all the show notes and our weekly picks for each episode. You can also listen to TWiV on demand on your mobile device using Stitcher Radio. It's a free app you can download, and you can use it on your iPhone, iPod Touch, BlackBerry, Palm Pre, and Google Android device to listen to TWiV on the go. TWiV's part of sciencepodcasters.org, promednetwork.com, and microbeworld.org, websites where you can find high-quality science podcasts and other science materials. Eric, thanks for having us today.
Pleasure is all mine.
Yeah, it's been great.
Great
And if you're ever in
talking,
San Francisco, look this guy up. You get a complete tour, right?
Beautiful.
You get the bridge and you get the viruses, the whole thing.
What more could you want?
Right.
It's beautiful. We really appreciate it, and I think our listeners will love hearing about what you're doing, and I love it. I think it's great stuff. Keep it up.
Thank you.
Thank you, Rich.
Oh, all the time.
Glad to have you out here.
Yeah, it's good fun.
As always, send us your questions and comments, twiv@twiv.tv. And next week, we'll be back to the usual stuff.
Geez, I won't be sitting next to you. I'll have to be looking at my computer again.
We'll do it over Skype. Maybe Alan Dove will be with us.
I would love to have everybody
But
back.
in a couple of weeks in July, we will be looking at each other again as we do this at the American Society for Virology. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.